Alrighty, I guess I'll just get started. How's it going, CannMed? Thank you so much for having me. It's great to be here. My name is Sam Haiden, and I am a research assistant at the University of Connecticut. I work in the Berkowitz lab, where we study the molecular genetics of flowering and trichome development in all of our favorite plant, Cannabis sativa. So, I am here to discuss with you today the mechanisms of short-day photoperiodic flowering initiation in cannabis, and also some of the implications that these mechanisms have on the phenomenon of the incomplete dominance of the photoperiod sensitive allele, and some of the impacts that that has on phenotype and flowering time. So, without further ado, let's begin. So first of all, I'd like to start off by describing the photoperiod loci. So I say loci because there's more than one locus that's associated with flowering, time, days to maturity, so on and so forth. So what is a locus? So a locus is a region of a chromosome in a genome which possesses genes which determine a phenotype, right? And so an allele possesses different versions of those genes on a homologous chromosome. So the photoperiod sensitive allele contains functional genes which respond to photoperiod and circadian rhythm. And these photoperiod responsive genes are dominant, right? So what does it mean to say that this gene is dominant? Well, it means that when the photoperiod sensitive version of this gene is in the presence of the photoperiod insensitive version of this gene, that the photoperiod sensitive will dominate the phenotype. And that's due to the molecular mechanism of the gene product. So we're going to take a look at one of the genes that's at the photoperiod locus, and that is PRR37. So this gene was described by Leckie et al. And Aurora Cannabis. And what you see in the top right is actually the ORF of the open reading frame of the protein. And this is the full-length protein from start to stop codon, right? And so this gene contains the molecular mechanisms that are required for the plant to respond to photoperiod. And due to the fact that it contains this molecular mechanism, when it's in the presence of the recessive allele, it will mask the recessive allele's phenotype, and that's because it possesses that molecular mechanism. So here we've got our full-length gene of PRR37, which we would find at the photoperiod sensitive allele, and here we have the photoperiod insensitive allele or version of this gene. So this is still PRR37, but it contains a splice site mutation in the gene, which results in a truncated protein. And so this gene is unable to respond to photoperiod and is the recessive allele. So what we end up with in this case are three different genotypes. We have our double dominant photoperiod sensitive individual at the top there. So this contains two copies of the dominant allele and is fully photoperiod sensitive, which results in a late flowering phenotype. So you can see the critical day length indicated by the sun there. So the full photoperiod dominant genotype is not going to go into flower until late in the season when the days are short, around 12 hours of critical day length, right? And so there in the middle we have our two recessive alleles, our double recessive individual, and this individual doesn't respond to photoperiod at all in any way, right? But this individual has an interesting special power that the photoperiod sensitive individuals do not have, and that is that these individuals can actually develop an inflorescence in response to age alone, which is something that's super fascinating because we frequently talk about what the autoflowering allele lacks in that it cannot initiate a response to photoperiod, but we rarely discuss what it's adding, which enables the plant to actually develop an inflorescence in response to age alone. So keep that in your mind for the future discussion. So at the bottom here, what we have is a bit of a phenomenon. So what we have down there is the heterozygous individual, which possesses the dominant allele as well as the recessive allele. And now the fascinating thing that happens when you have this genotype is that you end up with an early flowering photoperiod sensitive plant. So the plant is still responsive to photoperiod, but it tends to be much more sensitized to photoperiod. And so the critical day length at which this plant will go into flower or develop inflorescences is much longer. So we find that these individuals will actually go into flower when the days are 2 hours longer than their full photoperiod dominant counterparts. And this is a pretty significant finding for cultivation, because what that indicates is that these will develop inflorescences much earlier in the season, which is critically important when you're in a region which deals with mold or mildew or any of these issues that come with the later, colder stages of the season. So this is a pretty interesting result, which is very significant for breeders and developing fast flowering plants. Now, these right here, what we're looking at are diploids, right? So they have 2 copies of each chromosome, and what we're looking at is chromosome 1. Now, so this is the effect that we get when we have a diploid heterozygote. But when we have a triploid heterozygote, as was established by Drs. Lubell and Kurtz over at the University of Connecticut, is that when we have a triploid and we have a heterozygote with 2 copies of this autoflowering allele, we get an extra super duper fast flowering phenotype. And what this indicates is that the genes at the recessive photoperiod insensitive allele are dose dependent. So what that indicates is greater transcription of these genes results in a greater expression of this fast flowering phenotype. So the dose dependency of this allele was something super fascinating discovered by Lubell and Kurtz, and it's definitely not an insignificant find in terms of flowering time. And we can see that here in this graph, also produced by Lubell and Kurtz, where at the top we're looking at our triploid dominant photoperiod sensitive individuals, and they are beginning their terminal flowering at around 13 hours of daylight. Whereas the triploid heterozygous individuals with 2 copies of that recessive allele are actually beginning terminal flowering at closer to 15 hours, which happens much, much earlier in the season. And the way that plays out in the field is that what we're looking at here, I suppose if you go out into the field around mid-September and you're looking at your crop, that you're going to be much, much more pleased to see plant number 2 than you will plant number 5, because that plant number 2 has much, much more robust flowers than plant number 5 at the same time in the season. So this is a very significant phenotypic change that occurs. And it also begs the question of what is going on with this autoflowering allele that causes it to sensitize the photoperiod allele in such a way. So why the heck does that happen? This is a question that we set out to answer. And to begin, we thought we might ask, well, what are the mechanisms that induce the photoperiod response? Also, what are the mechanisms that induce the autoflowering response? And how do these 2 pathways not only respond to their environment and to photoperiod, but to one another? How is the crosstalk occurring, which enables this autoflowering pathway to sensitize the photoperiod pathway? So, as with all things in plant biology, we begin to answer these questions by taking a look at Arabidopsis. So this is a illustration of all the different flowering pathways that occur in Arabidopsis, but we are focusing on the photoperiod pathway down there in the bottom left. So this pathway possesses a host of highly conserved genes which exist across plant species named Gigantea, CONSTANS, and FT, or florigen. So these 3 genes function in response to photoreceptors, namely Cryptochrome and Phytochrome. So these are molecules that exist in the leaf, and when they're irradiated by certain wavelengths of light, they become active, and they go into the nucleus, and they begin to activate certain transcriptional changes. There's a whole host of transcriptional changes associated with the phytochromes, but we in particular are looking at these photoperiodic flowering genes, right? So in Arabidopsis, what happens is that during long days, these photoreceptors entrain circadian rhythm genes like gigantea, which is a circadian rhythm gene, as well as CONSTANS, which is in Arabidopsis is a central integrator of different signals. And what happens is that gradually as the days increase, the expression of CONSTANS increases, and along with the expression of CONSTANS comes an increased expression of this gene FT. And that's because CONSTANS is a transcriptional activator of FT. So this is a pretty well-established pathway in Arabidopsis, but Arabidopsis is a long day plant. Arabidopsis goes into flower under long days, so it's very different from Cannabis and other short day plants. So the difference in particular is displayed here. So this is a model from rice, and it may seem inappropriate to apply a model from rice to cannabis due to their obvious unrelatedness. But interestingly, we've actually found a significant number of sort of homologous mechanisms and homologous genes between rice and cannabis, not the least of which being this gene PRR37, which we mentioned before, which possesses this molecular mechanism which responds to photoperiod. And due to the fact that it responds to photoperiod, it's critically day-length gated, right? And what that indicates is that it has a biphasic function, right? So it has one discrete function during long days and a completely separate discrete function during short days. Similarly, so does CONSTANS. And we've been able to see that this sort of pattern is present in both rice and cannabis. So I would say that it's not entirely unreasonable to suggest that this mechanism is likely conserved between rice and cannabis. So we're proceeding forward using this model as the most likely way for short day photoperiodic flowering initiation in cannabis. And the way that this works is that there are one or more genes expressed in the leaf whose protein products actually directly interact with the CONSTANS gene. And that prevents CONSTANS from performing its function of upregulating FT. So that's how things are working during long days. During short days, these circadian rhythm genes are affected and the expression patterns of these repressors fail. And what happens is they release their repression of CONSTANS, allowing CONSTANS to go ahead and upregulate FT. And this is a critically day-length gated biphasic function. So as a result, we thought that it would be wise to start trying to identify critically day-length gated genes, or genes which have significant expression changes based on photoperiod. And then secondarily, of course, we want to see whether or not these critically day-length gated genes are interacting with CONSTANS directly in any way to repress or initiate flowering. So this is more or less the model we've been working on and the model that we're continuing to study and to test. So here's a little illustration of how that model, of this putative model, and how it is likely to work in the cannabis plant. So here we have our happy plant, vegetative, long days. We have CONSTANS, which exists in the leaf, and it is being repressed by these interactors due to the fact that we are under long day conditions. So everything's hanging out, just being very vegetative, not very exciting. But then, upon the transition into short days, what we find is that these cryptochromes and phytochromes are entraining these circadian rhythm genes, which are also entraining the CONSTANS gene, and the result is the failure of the expression patterns of these repressors. And when these repressors fail, they allow CONSTANS to go forth and perform its function of upregulating FT. FT, florigen, is expressed in the leaf where it translocates up to the meristem and activates floral meristem identity genes like SOC1 and leafy, which result in beautiful trichome-laden flowers that we love so much. So this here is sort of the putative model of what's going on in the cannabis plant with regards to photoperiodic flowering initiation. And so the photoperiod genes at the insensitive allele, the autoflowering allele, do not elicit a response. So they don't do this part. They fail to respond to photoperiod to modulate the activity of CONSTANS and therefore FT. So they're not doing that, right? But now here's the question that I'd like to pose for all of you, because I don't really see anybody asking this question. How is it that the insensitive allele is causing this phenomenon of incomplete dominance? How is it that the insensitive allele is actually modulating the photoperiod response and widening the critical day length in heterozygotes, resulting in a fast flowering phenotype? I think that's a question worth asking. I don't have answers yet, but we're working on it. So probably one thing to remind everybody in the room about before we continue discussing the subject is that cannabis actually does produce solitary flowers during the mature vegetative phase, right? So it doesn't actually require short days to flower. All cannabis genotypes, upon entering the mature vegetative phase, produce single solitary flowers, but they don't produce inflorescences. So what we're really talking about when we say short day flowering is the development of the inflorescence. And photoperiod sensitive plants simply will not produce an inflorescence unless you expose them to short days. It's impossible for them to develop an inflorescence based on age alone. However, in our autoflowering individuals, we see that they obviously have that capacity. They can grow to a certain age and then develop an inflorescence. So we constantly talk about what these autoflowering alleles are lacking in terms of their ability to respond to photoperiod, but we rarely discuss the additive function that's coming from the autoflowering alleles. And I think that that's a really ripe subject of study to be looking into. So again, it's also it's worthwhile to remember that only one autoflowering locus has been well characterized, but there are at least 3 other loci in the cannabis genome, which were described and labeled DTM 2, 3, and 4 by Steele et al. At La Trobe University. And DTM2, which is on chromosome 2, contains a host of SPL genes, which is a family of genes which encode transcription factors, which activate transcription. And these genes tend to activate SOC1, which, as we learned before, is our floral meristem identity gene. It's a master transcriptional regulator. So effectively, what we're seeing is another pathway to flowering, which is, of course, expressed in all cannabis genotypes, but only seems to lead to age-dependent development of inflorescences in autoflowering plants. So I do believe that this would be an interesting place to look to try and understand what's going on with this phenomenon of incomplete dominance. So in the event that you're a bioinformatician and you want to take a look at what genes are at these loci, here you have it. This is a little map of the chromosomal locations of DTM 1, 2, 3, and 4, where 2, 3, and 4 are largely uncharacterized. And here would be sort of like a graphical illustration of what might be happening in these fast-flowering, incompletely dominant individuals, right? So on the right side here, you're seeing the photoperiod pathway. What you're seeing is the reaction of these repressors of CONSTANS to the entrainment of circadian rhythm genes during short days, activation of CONSTANS, activation of FT, floral meristem identity. But then over there on the left, you're seeing a less talked about pathway. And this is the pathway that induces solitary flowers in vegetative plants, but it is also more than likely, maybe some minor differences in these genes are actually contributing to the modulation of the photoperiod pathway. So what we're proposing here is that this age-dependent pathway, which depends on the expression of a couple of microRNAs, which regulate SPLs, which regulate SOC1, that this pathway in the autoflowering individuals might actually be having an effect on the photoperiodic response, modulating it and sensitizing it, which makes it result in this fast flowering phenotype. So this is sort of the best model we have to work with at the time. But a good question also is, how did we get here? Where did this begin? Because I'll tell you, we didn't start by looking into incomplete dominance of the autoflowering allele. If you remember my talk from last year, what we discussed was the use of spectral diet to try and modulate gene expression and also biomass accumulation and cannabinoid accumulation. We were trying to see if we could modulate red-far red ratios and therefore affect Phytochrome, the photoreceptor I discussed, which would affect gene expression and induce changes in the phenotype. Disappointingly, we were not able to affect a change in biomass or yield or any other sort of phenotypic measurements that we took. However, we did find that we were able to affect the expression of genes in the photoperiodic flowering pathway, which indicates, of course, that they are responsive to Photochrome. And in addition, we basically found that we were able to use this sample set to create a transcriptional map of the changes that occur in the photoperiodic flowering genes, which results in photoperiodic flowering, because we had samples from the leaf from pre-short days, as well as 4 different time points after short days. And we also have the meristem tissue from the end of week 1 and week 2. So in doing this, we were able to sort of, map the transcriptional changes that occur in the leaf, which result in signals to the meristem, which result in further transcriptional changes. And interestingly, we did start to see some strange results. Almost nothing makes sense, but that's a lot of fun as a scientist, you know? If things don't make sense, we want to find out why. And one of the first things that don't make sense is that we were able to increase the expression of CONSTANS significantly by modulating Photochrome activity. But the strange finding is that this increased expression of CONSTANS was correlated with a decreased expression in FT, this floral activator. And knowing that the CONSTANS gene is supposed to be a transcriptional activator of FT, it really doesn't make any sense that its increased expression would be correlated with a decreased expression of FT. So that's the first strange finding. Followed up by 2 expected findings, which were good. We found that PRR37 expression decreased significantly upon the entry into short days. And that makes sense because PRR37 is putatively a repressor of that floral meristem activator gene FT, which would, of course, be degraded upon the entry into short days, after which it allows CONSTANS to go ahead and activate FT. So this further confirms the function of PRR37 as a biphasic regulator, which also sort of lends to the notion that this rice model could be an effective one for cannabis. And then, of course, we see SOC1, our floral meristem identity gene, which is significantly upregulated by day 14 following the activation by florigen. So, having found these results using qPCR, we decided to go ahead and take it further and try to identify some more critically day-length gated genes by using bioinformatics. But prior to that, we did some more qPCR on the CONSTANS gene, which of course we've been discussing at length. Now, the CONSTANS gene in rice, the expression of it doesn't change. Its activity is more or less determined by the presence of these interactors, right? So it's been shown that CONSTANS expression in rice doesn't change very much. In Arabidopsis, CONSTANS expression during the flowering period tends to increase, right? During long days, CONSTANS increases, it increases the expression of FT and leads to this gradual development of flowers. So there are 13 different CONSTANS genes in the cannabis genome, and we identified the ones that were most highly expressed in our wife cultivar. And we studied the ones that were most highly expressed, and we found that the majority of the CONSTANS genes were actually downregulated upon the entry into short days, which, given that it is generally acting as an activator of flowering, this doesn't line up. It's a little strange. One of the genes, CONSTANS number 9, did not change, and we were able to find that only one of the CONSTANS genes, CONSTANS number 5, seemed to be significantly responsive to Phytochrome. And that's also interesting because CONSTANS number 5 is the QTL that was given to us by Austin Shep at Rapid Genomics. So more than likely, we're going to continue looking at CONSTANS number 5 as the potential activator here. So now we're getting to the transcriptome that I mentioned previously, the bioinformatic aspect of this study. So we took our leaf samples at pre-short days or day 0, and we took our leaf samples from the same plants at day 7 of short days. And as you can see, obviously the critical day length is completely flipped from those two sample types. So we took the total RNA from these leaf samples, and we sequenced the mRNAs, which results in what's called a transcriptome. And we took the transcriptome and performed a differential expression analysis. And the goal of this is simply enough to see which genes were up or down regulated by the entry into short days. And what this is going to do is give us a list of CDL-gated genes, which are potentially involved in the transition into flowering and may also be interacting with CONSTANS to perform this function. So thankfully, I was able to find a really brilliant bioinformatician to help me out with this. His name is Nathan Johnson from Vern Bioanalytics. I'm very grateful for him. And we were able to produce a differential expression analysis, which gave us a very long list of genes both down and upregulated upon the entry into short days. And so what this is giving us is our list of CDL-gated genes, which we can continue to study to look for other interactors with CONSTANS, which are sort of mediating this transition into short days. And I would also like to mention that we mapped and annotated these transcriptomes against 9 different reference genomes and got coverage ranging from 10 to 70%, where the Jamaican lion genome was easily by far the highest mapping and annotation at 70%. So we were really grateful for that complete genome to use as a reference. So, moving forward, where it's headed, we would like to actually take PRR37 and demonstrate that it does directly interact with CONSTANS to activate FT, as the model would suggest. And we would also, of course, like to start studying these other CDL-gated genes, which are homologous to rice genes, which may also be interacting with CONSTANS to have some sort of effect. And additionally, any gene that's modulated by critical day length may also have some sort of function that it's performing in the plant. So yeast 2 hybrid, and then of course we need to dig into the genes that are at the autoflowering alleles, not the least of which including these microRNA species, which I didn't discuss very much, but are more than likely critically important in this process. And of course also SPLs, which is this whole host of less studied genes in cannabis which activate SOC1. So that's where it's headed, and that is my talk. I'd like to give a big thanks to Dr. Gerald Berkowitz for his continued support, as always, especially to Dr. Nathan Johnson from Vern Bio, who was so gracious to help with the bioinformatic work, and definitely Austin Shep from Rapid Genomics, who provided us with the list of QTLs we used for our qPCR analysis to begin. So thank you very much. I'll take any questions.
Nice talk.
Thank you.
Do you have any evidence that nutrition could affect the transition to flowering? I have seen several nitrate-responsive genes, SPLs, and things like this. So do you have any data showing that nutrition could delay or accelerate the transition?
Could delay or accelerate what exactly?
The transition to the flowering process.
The transition to the flowering process. Nutrition, we don't have any data specifically on the effects of nutrition affecting gene expression or anything like that, but I do suspect that, you know, obviously having optimum plant health is going to optimize this transition into flowering, but we don't have any data specifically on how nutrition would affect that, no.
Thank you.
Yeah.
I just had a quick question for you. So how could this information help smaller co-op farmers and growers that are looking to, you know, get into this industry that is very big dominated, but also with this science and bringing it in and giving their plants that really good chance of producing excellent quality?
Oh yeah, well I think this would be critical for that actually, because, you know, one of the major barriers to growing cannabis is sort of investment in the facilities that it requires. But if you're able to grow plants outdoors successfully, that would obviously be the least expensive way to get started. And you know, many growers in northern latitudes in particular are limited as to what they can grow outdoors because the plant has to finish, or else they won't be able to harvest it in time to actually, you know, collect a harvest. So it's critically important for for breeders in particular, to develop fast flowering individuals for the success of farmers generally, but in particular those who want to be cultivating outdoors, which is probably the preferred way in terms of, you know, environmental sustainability. Of course, we don't want to be growing underneath lights like that, right? And so having a fast flowering individual definitely supports small farmers and organic farmers in particular, and being able to grow in the field at northern latitudes in particular. Yeah. Okay. Thank you very much.